Hi, welcome to the Cannabis Safety Testing Panel. My day job is Director of Clinical Research at Tilray, but I also serve as a member of the Cannabis Advisory Committee to the state of California, advising them on their regulations in California. And this issue of product quality safety is really maybe not very interesting to a lot of you, but it's actually critical to elevating this industry, and it's a very difficult topic to cover. So I have with me here 4 chemistry experts, and they're going to help us kind of discuss this topic and tease apart some of the most important issues that we need to be thinking about. And I'll let them introduce themselves.
Hello, my name is Khail Pasion. I'm representing MedMen Enterprises, LLC. We own, manage, and operate a slew of licenses for the cannabis industry across the nation. I serve as their laboratory director for compliance, specifically ISO 17025 for our in-house analytical testing facilities that are built within our facilities. My background is in biochemistry and laboratory diagnostic assay development, and I am coming here by lieu of Doug Kennedy to speak to compliance and regulations for analytical laboratories in the cannabis industry.
My name is Khail Boyar, and I'm a field application scientist with medicinal genomics. I received my bachelor's degree in neuroscience and behavior from the University of California, Santa Cruz, and I currently serve as the vice chair for the American Chemical Society's Cannabis Chemistry Subdivision, as well as their scholarship chair. And my background is mostly in cannabis testing. I've been doing it for about 6 years now. And I got my start at one of the first independent analytical laboratories in the state of California. And in those 6 years, I've tested about 100,000 samples of medical cannabis at this point.
Hello, everyone. My name is Andrew Pham. I currently serve as the scientific director at Bell Costa Labs, the first licensed and accredited lab in Long Beach, California. I am also the chair of the Cannabis Chemistry Subdivision at the American Chemical Society, as well as a court-qualified expert witness testifying in over 24 cases involving cannabis.
And I'm Mike Flournoy, President Eurofins, Frontier Global Sciences. Been in this analytical world since 1989. I feel pretty old. But been really grateful and going through all of the cannabis documentation and with all of the sciences going on with it. And I'm here to hopefully help navigate through some of the quality pieces, because this is a really infant type of industry, and there's a lot of information that can be learned from the past that we're hoping to put together with this.
Thank you. Okay, so I'm going to set the stage a little bit by telling you what I currently see as a cannabis, in the state of California. So full disclosure, I have a son with very severe treatment-resistant epilepsy and have been looking for products that are safe to give him for the last 6 years. Even today, if I walk into a dispensary, I'm not comfortable with the product quality standards of any product that I find in a dispensary, at least not in where I live in Northern California. I don't know whether the product will be contaminated. I don't know whether the potency is correct, right? Whether the labeling for THC and CBD content is accurate. And I think this is a real problem. And I'm a very educated consumer because of my background. And I think we're at a place where a lot of consumers are not as educated and really don't understand that nobody is overseeing these cannabis products being sold in dispensaries. Because right now, it's the industry itself that is kind of paving the way. And one of the things that I wanted to talk about with you guys today is how do we ensure that the testing industry, with lack of federal oversight, and I think the speaker from medicinal genomics gave a good overview of how the regulations differ state by state and how the states apply different standards or they reference different pharmacopoeias to establish their regulations. But if there's going to be no federal oversight, how can we as an industry raise the bar in testing to make sure that a sample that is being given to 2 different labs in, let's just say in California, would return the exact same results. I mean, that's the goal that we're going for. And so let's start with Khail. You. Yeah.
2 Khails.
What are some of your suggestions for, for the testing industry itself to raise the bar? I know you guys are all 4 part of different organizations, and maybe you can speak a little bit to what the different organizations do and which one might be the best suited to set, third-party standards for all of the testing labs.
So for my case example, we're not an independent laboratory testing facility. We do have laboratories within our vertically aligned facilities. And with that being said, we have to take into account local jurisdictions across many different states. From my background in both pharma and biotech and even in clinical, we follow strict regulations, specifically ISO, International Standard Organizations, and even more so specifically ISO 17025. With my experience going from state to state and dealing with local jurisdictions is that a lot of the authorities there, for example, fire marshals, if not county authorities, rely on people like us, people in the industry, to educate them on how to build a regulatory system, how to apply that on us. So in terms of moving the compliance and regulation forward to make sure that it is moving towards a federal legalization. At MedMen, we impose local jurisdictions and local standardization and also ISO as a prerequisite for a lot of our facilities and a lot of building analytical labs and also developing more methods. It also speaks to teaching a new generation, especially with this new version of ISO 17025, which takes out a lot of the details from 2005 and leaves a lot of things generalized. But in a sense, this opens a door for innovation to allow more creativity that still follows compliance and regulations. So with that being said, it gives an opportunity for perhaps legacy growers or people who have been growing in things for 20 years in their garage who say that they can honestly grow this with a certain distance or whatnot, but enable to teach them to use objective evidence, like using an analytical lab to actually track and monitor chemicals and whatnot. So it's important to use regulations and compliance to not only help this industry move forward, but to also help the people within the industry to be more familiar with the progression of regulation and compliance.
Okay, I'm going to ask Michael a question because you come to this with background from other industries, so environmental testing, food testing. What are some of so given your extensive background in these industries, which are well regulated, what can we take from each of those regulatory standards and apply to this industry?
And you raise a really good question. And technically, this particular chemical, this particular plant, this particular food, it could be, transcends a lot. It's not a drug. Actually, it is a drug. It's not a food. Why should you put it in food? It's really not something that it's like a tobacco, but it is because you can smoke it. So it goes across many different markets, many different fields that really puts it into a different category. So you don't really look at it as a drug, but FDA is there, DEA is there, and they do look at it as a drug, but we don't. The DEA really puts a clamp down even on the DOT, so being able to move things across really makes it very, very challenging. And now, kind of looking back at the past, working with the EPA and the FDA, back when they were developing methods back in the 60s, 70s, 80s, it was kind of like the Wild West, kind of like it is today, specifically on the analytical side, on the testing side. If I recall, we were able to do one-point calibrations back then. We were able to hold the calibration for as long as we wanted to. All of those changed because it was a very big need for getting accurate data. So using some of the lessons learned from those industries from the past, we can certainly use that information today to make this particular process much faster to the endpoint as possible. And being able to, kind of distill everything that we've done, it all comes down to validation. That's what it all comes down to. So if you're able to get a good validation protocol with the intention of having good limits, because that's also part of it, that will definitely get you to a point where it starts defining what type of instrumentation you'll need, what type of chemistry analysis you'll need, what type of processes you'll need to be able to make things move forward. But it also gives you an opportunity to be able to let technology help you grow faster, help you get better over time, because if you start to constrain everything because you've made it too constrained through your methodology, then it's really hard to help technology grow. So I think that's kind of the gist.
Basically striking a balance between allowing innovation to occur in a very nascent industry, but still superimposing some of the standards that are common in other industries. And one example for the audience is common reference standards don't exist, right? So a reference standard is by definition the same thing every time any lab uses it. And currently in California, there is no reference standard for any of the cannabinoids, which means that you can't call it a reference standard, right? And Andrew, a question for you might be, what has your experience been, serving customers in what are some of the issues that have come up because of the lack of a common reference standard?
Well, it's the same complaint that pretty much every one of our clients is saying. Well, we're a distributor, we take a same sample to 10 different labs and we're getting 10 different results. And there are so many technical difficulties to overcome in order to fix this, but yeah, an actual common reference standard would be extremely helpful in really getting everyone on the same page. It's honestly, in my opinion, if this can't be done on the federal level, I think the burden is actually on the state. States have established reference testing laboratories. So if the state has a reference testing lab, they can act as the referee. You can present your validation data to them, and then you can actually compare your data to other labs who anonymously. And I really think that's really the best path forward, because the state reference testing labs won't really be motivated by churning out results in order to make money. The resources are going to be from tax revenues. So they'll actually have resources, they'll have equipment in order to really get these methods together down and be able to share these with the testing labs that are doing this commercially.
That's great. Another problem we see is that people use different equipment to test products, right? And the equipment has to be calibrated a certain way. It has to be cleaned a certain way. These are all called standard operating procedures. And so I'll give this question to Khail Boyar. What has your experience been when you see the different types of equipment being used in different labs, and do labs have standard operating procedures in place that are relevant to the types of equipment that they use? And how can we standardize that process a bit more? Would it come from an external regulatory body? Would it come from the manufacturer of the equipment? Can you speak a little bit to that?
Certainly, yeah. And I think really what it is we all need to agree on, what is the appropriate instrumentation to quantify or detect a given analyte or, I guess, in the case of microbiology, pathogens or species of interest. And really what it comes down to is the states need to all come together and establish some sort of consensus as far as, okay, here's what we need to be looking for, and then here's the appropriate methodology to do it. A good example of this that I see is cannabis microbiology. My colleague Yvonne spoke to this quite a bit. We have a kind of scattered mismatch between the states. Some states are doing total count tests. Some of them are doing species-specific pathogen tests. Some of them are using plating. Some of them are using quantitative PCR. And all these systems, they vary in their results and their methods. And I think really what it is it's hard to establish any type of consensus when there's so many people doing so many different things. And so I think it needs to come from all these labs. They really need to come together and collaborate and speak with each other. And all these states, they also need to collaborate and speak with each other. What do you feel is the most important in terms of patient safety, and what levels are appropriate? And then dictate, what methodology is used and what type of instrumentation is used. And as of right now, I don't think that there is that consensus. Right now, we're all scattered doing our own thing, and I think California is doing a good job of paving the way for establishing some consensus because they've really done a lot of diligence in figuring out what is important for patient safety. And I think that, we just all need to have this conversation in order to arrive at what is the appropriate methodologies in order to keep patients safe.
So let's go back to, so I think we've talked a lot about the problem, and it's a huge problem, and I think it's going to take years to address, but let's focus a little bit on the solution. So you have mentioned different societies that you belong to. Which would be the third party that could take on establishing, SOPs or common testing methods in the cannabis industry that are specific to the cannabis industry and address some of the issues that we see that may not be seen in food or pharma or environmental arenas?
One of the bigger areas that's doing there's a lot of science going on, a lot of research going on. AOAC is working really diligently on developing methods that could do a majority or a large amount of the testing for different products. And being able to validate every product is almost impossible. But there's certainly ways you can group things into categories, and if the AOAC and some of these other organizations can kind of push that.
Sorry, Michael, what's the AOAC?
It's the Association of.
It used to stand for Association of Analytical Chemists, but recently they just changed it to AOAC, so now it just stands for AOAC.
Yeah, it's just simple.
Yeah.
But looking at just the methodology, they're putting together a set of criteria, a set of quality criteria to be able to do the process of digesting or extracting and analyzing samples for results, to be able to mitigate interferences and be able to mitigate the processes so that you're getting better quality data and not having huge biases one way or the other. But again, there's lots of different areas, and like I said, this is actually going to transcend just one market. And right now we're looking at it as more of a food, drug type of thing, but it's going to get environment. It's going to get into the environment, and we're going to have to test it in the environmental space. So we have to start looking at exactly what that means, and then that'll go into more EPA-style methods, and it'll be in the permits and all this other stuff. So, kind of looking at it from a standpoint of what do we need to do as an organization, we as a community, we have to really engage with some of the organizations that you may belong to so that we can try to come together on some of these things. There's other advocacy organizations that are out there, ACIL, the Association of Association of, yeah, there you go, thank you. And I'm on the board too, what the heck. But anyways, but looking at some of these other organizations that are out there, we should all come together and be able to help the government be able to put together something that we can all live with. If we go back to the past, that was when the EPA developed the methods that the labs were supposed to use. And it worked for a while, but it didn't work later. Same thing with the FDA. They developed these methods, and then the labs were supposed to use those methods. Well, it so happens that some of these companies that are doing the testing are pretty darn smart, and they're able to do a lot of these developments on their own. So, kind of looking at how we can put all of this together, we all need to come together as a group and almost demand. We have to kind of demand on what we think is going to be best for the consumers, because the reason why we're all here at the testing level is to make sure that we're, making our communities safe. At the EPA, it's community safety. FDA, community safety. Even DOT, community safety. So, at every level, this is all about safety for consumption, safety for the community, safety for our children. So I think we all need to come together and put together something that's going to work for this particular product.
Right, and so you brought up something important, which is this comes in a lot of different forms. Like a gummy bear could come to the lab, a brownie, a candy, and dissolving all of those and then being able to run a test that's accurate and coming up for a method for all of these different ingredients is going to be difficult. So where do we? So I see, the end result, what the ideal would be. But if we're going to tackle it one step at a time, what are the most important things to test for right now? I mean, we heard that certain forms of aspergillus are much more harmful than others, and currently very few people are testing for those. You know, what's the first step in looking at safety? What would be the very first step and the minimum standards? And then we can go from there.
Yeah. So I think when it comes to, at least with microbiology, you definitely hit it with aspergillus. I mean, this is like it's an endophyte in the cannabis plant, and it's commonly found, and it's one of the actual pathogens that can kill people. That it's well documented that it can kill people. And when it comes to pesticides, I think we need to understand, okay, what is commonly being used, and at what levels is it actually okay to have them in. For example, things like myclobutanil are constantly being used, and there's, of course, some research that's been done to show, you know, that it produces cyanide when it's combusted. So that's clearly an unsafe product for any type of smokable product. But you know, what is being commonly used, and establish these limits based on science. I think that's what's currently happening right now. Is no one's really basing these regulations on scientific research. They're just taking their best educated guess as to what is going to keep people safe. But that also puts people in the testing labs in a really tough position, because you set the bar or your detection limits extremely low, and what's allowable at extremely low levels, it becomes very difficult to actually comply with these regulations, because again, it's not based on science, but it's based on what they feel is keeping people safe. So I really think that a lot more research needs to go into what is an acceptable level of certain contaminants in cannabis. Of course, there's also heavy metals as well that need to be tested for, and of course, that's something that, you know, we should be absolutely testing for all across the board. And I think no levels of heavy metals, you know, or very low levels are going to be tolerated, but we need to establish consensus as far as what is okay and what is not okay. And then as far as mycotoxins go, that's another contaminant that at the moment, I just don't think we have enough data to support, you know, how rigorously this mycotoxin testing needs to be done. Of course, we see that there is aspergillus there, but are the conditions sufficient enough to produce these types of mycotoxins that could be deleterious to human health? I think that it's just, there's a lot more research that needs to be done in order to establish what these limits should be. And right now, we're just taking our best educated guess at it. But I think we need to see more data in order to actually establish proper limits.
I have one thing. Way back in the 60s, 70s, 80s, they did lots of studies that determine what type of chemicals, specifically in the environment, I'm kind of going back, were going to be harmful at what levels, which is how they developed these permit levels. I think we do need to do that for these, because, you know, at the contaminant level, you're absolutely right. You know, do you have to speciate something to be able to determine which one is going to be harmful? For example, arsenic. Do you have some forms of good arsenic? There's some forms of bad arsenic. The bad arsenic is really, really bad. You don't want to take that. But the good arsenic, you know, you can take quite a bit of it and you'll be fine. But as these limits are progressing, you have to be able to determine what is going to be important to the consumer. I just heard today that, you know, from a friend I just met, that North Carolina is using an an instrument that is not very capable of being able to determine which is the bad portion of a chemical that is trying to do, and that's, you know, basically THC. And THC is not really that bad, but you're kind of looking at how much of it is going to be the right amount is going to really determine what type of instrumentation you're going to be able to need to do that particular analysis or the species of that chemical. So I think there needs to be some studies on what it is that we're going to be calling important, and understanding that that's going to change, because over time you're going to have different technology that's going to be able to do other things. But, you know, being able to determine what is going to be important to the consumer is super important. You know, some of the levels that I've seen just in California, you know, to me, those are high numbers based off of the type of testing that we do. You know, for example, for arsenic is, you know, 0.1 and for 0.1 ppm, and then for mercury is bad, but there's, you know, some mercury that is not horrible. But methylmercury is super bad for you. And if you have 0.05 micrograms or milligrams ppm of methylmercury, that's not really good for you, not good at all. So, you know, we have to come together with what these type of limits need to be based off of the science. Absolutely right. And just because it's called arsenic doesn't mean that's part of the science, because there's many, many forms of arsenic. So I think that's the important part.
So I'll just throw this out there because I don't think either of you answered my question, which is, what do we start with? So we know that a lot of research needs to be done, but we need to start somewhere. So what do we start with right now?
I really think it starts with toxicological studies, basically making sure and studying exactly how much. To me, it's pesticides is the largest public health danger right now because we don't have a great idea on how much is dangerous to human health in terms of inhalation routes. Obviously, a lot of these pesticides have been used in food for decades, but we don't really know how dangerous it is when you put it to a flame and inhale it. So I think that's really what's important here. I also want to bring up heavy metal testing is obviously going to be important a lot. Now that a lot of these vape carts are being filled with oil, the carts themselves are being produced from China, which doesn't really have a lot of heavy metal testing. So contamination levels that we're seeing in vape carts, I mean, there's just not a lot of data right now, and I think it's an extremely big risk.
Well, it's interesting that you bring up vape cartridges because they're putting substances in the vape cartridges like PEG, and we don't know what the long-term risks are of inhaling PEG. I mean, it's not a good chemical, and there's nobody overseeing that anywhere, which is really scary to me. So that's a separate issue, though. So if we were to put in place certain standards right now, I mean, clearly we need to know accurate potency, right? Because we want to avoid overdosing. As far as contaminants, what would be, so the 0.1 parts per million for arsenic, Michael, is, I think that's in the AHP and the USP, and it's okay with them, but now I'm hearing you say that that's too high.
It's pretty high to me. That based off of some of the food studies that we do. Once you get into, Prop 65, I want to bring that up, Prop 65. Once you get into serving size, people are eating grams and, mini grams of this stuff. It adds up pretty quick. That's why you need the C lower. That's the reason.
Right. So, I mean, that's actually pretty scary to me because that's the standard that almost everyone everywhere follows, the 0.1 parts per million for arsenic.
Yeah, we look down to 0.001.
Okay.
That's our MDL.
Okay. So in Canada, we can't use pesticides because the government can't regulate the pesticides, right? They haven't decided what's an acceptable pesticide to use on cannabis, so we use none. Here, the EPA has not really put out a list for which pesticides are okay to use and at what levels. So does that mean we should not be using pesticides at all?
I don't think so. There's some naturally derived pesticides that have definitely been readily widespread and available. I think even California put out a list of acceptable pesticides, did they not?
California did.
Yeah, so we're leading the way.
Okay.
I have something to contribute to
Go
your
ahead.
previous question of where should we start, and I think it follows back to Mike's answers. We should just all start with validation, proper, full validation, because there aren't any standard methods available to use and to be accredited for in the nation. And so we have to develop our own methods in-house and get those validated. And a third-party body to oversee that would be going for accreditation. To get accreditation, to get an international standard accreditation, you have to go through an accredited body. That accredited body is a part of a bigger national body that oversees competency, that oversees accuracy, precision of labs, of testing labs, including cannabis labs. And so if we start with a robust validation program for any of our methods, pesticides, heavy metals, even potency, and then a part of that validation is also a robust proficiency test, and that speaks back to your certified reference materials, the need for that, then that would get the ball rolling with all of us at least measuring the same thing the same way all together.
Right, so you're talking about proficiency testing, which the state could even oversee, theoretically, because you're allowed to develop your own methods. You just have to show that you get the same result every time you use your methods. But for the so how many of you have a Schedule I DEA license, or do you need one in order to get there are commercially available reference standards, And are you using those? Do you see anyone using those? Do they require a Schedule I DEA license?
Yeah, I think you pushed the button.
So there are actually commercially available reference standards that you don't need a DEA license with as long as it's in a concentration of 1,000 milligrams per mil or less. So that's what most of the testing laboratories are using right now. There's also, the Emerald Scientific has also started putting out reference standards that are in a hemp bud matrix. So basically, if it has less than 0.3% THC, it's not technically unlawful to ship across state lines, although it's a really big gray area. But I think that's a really good step forward because proficiency tests in the past have basically only just been these vials of reference standards at certain concentrations. And at that point, you're really just testing at where your machine is. All you have to do is dilute it and put it in. But it doesn't evaluate your sample preparation method at all, which is probably more than half of the reason that there are so many disparate results among labs. Not only that, sample collection is a really big deal. If you're not collecting the sample in a statistically valid and representative manner, then you're not going to get the same result.
So we have about 10 minutes left, and I think I'll open it up to questions.
Hi there. My name is Diane Strauss, and I'm a member of the Hemp Roundtable. I'm not sure if you guys are familiar with the Hemp Roundtable or not, but the Hemp Roundtable has put forth a great amount of resources to produce a hemp certification program that has been created by Where Food Comes From. Where Food Comes From is an agency that developed the USDA organic standards and has developed many organics and many other standards and certification programs for a lot of the food industry. And it is going to be validated by Validus. They are going to be the third-party audit on this program for hemp producers, growers, and processors. It is going to be unveiled at HIA in Los Angeles. The HIA show starts, I think, on November 2nd or 3rd of this month. And it does address a lot of the things that that you guys have been bringing up. And in the hemp industry, we felt that it was imperative that the industry itself started setting standards so that people could have confidence in safety in the testing and in the processings of which in way things are being produced and being offered to the public. So soon you will be able to go. It's not quite up yet, the URL and the website for our landing page to see the standards. I'm sure that they would love to get feedback from many people on the certification method that was created. It will be an ongoing and an improving certification program and will be available January 1st.
And what was your name again?
My name is Diane Strauss.
Diane Strauss.
And it'll be, I think it's going to be, it's ushempauthority.org. ushempauthority.org will be the URL that will be, or you could also go to the US Hemp Roundtable. I'm sure there'll be a link, but you hopefully in those standards can also be used in the cannabis industry, meaning the THC industry as well, even though this is specifically certifying to show that it is less than 0.3 and it has the GMP standards that are going to be needed from grower all the way from seed to processing.
Thank you.
You sure?
Hi, my name is David Schur. I own a specialty chemical company dealing with chromatography. So we work with, you want to know about the process and the best way to deal with that. I think it's the stationary phase people that make sample prep products that you need to work with, those who make SPE cartridges, those who make the analytical instruments such as Shimadzu, and working with them to define the best methods for sample prep, because that is right now one of the biggest problems that we've seen in trying to determine proper concentration of pesticides, et cetera. But my company right now is defining a true method for decontaminating pesticides from cannabis extracts. So we're currently in an alpha stage with that and currently going to a beta stage. So I'd love to speak with any of you to talk about that. But right now, you've made a good point. You've got the highest quality of analytical testing equipment and consumables for you guys to do the best work. The challenge is how to successfully use them. And you brought out a great point about sample prep. I mean, right now, we're sending samples to different labs, the same sample getting different results. So when you have that, just as something for pesticides, besides concentration of THC, CBD, and other compounds within that, it is a challenge. But I go to the people that make chromatography products. We're one of them. So we can help you in defining those methods. So anyway, I just wanted to bring that up. But there is a process that we're making for you to understand for total decontamination of pesticides from the extracts while maintaining the integrity of the cannabinoids. So I'd like to talk with you guys after. Thanks.
Absolutely.
Thank you.
Hi, thanks guys for mentioning method validation. I think that's really important in the interim until we get some actual standards in this industry. But I had a question about if you see any usefulness in round robin testing as a way to compare labs either within a state. I guess it's difficult to do that across states at this point. But could you just comment on the role you might see or not see for round robin testing?
I think it's great. Round robins are a great exercise. It really shows deltas between different results. And kind of going back to his result or his response on getting good sample prep, doing the pesticides or even something as simple as metals. If you have an oil, you have to get it into like an aqueous form, and being able to do that is not very simple. But yeah, I do think there is something there. You probably have to have someone who's run round robins before to do this. The trick is that you have to somehow do something similar across state lines and then be able to bring all the data together. Or if you're okay with just having a state round robin, I think you have to kind of make that decision if you're okay with it. But to me, I think it's great to be able to do that. The good thing about the hemp thing, though, is if we're able to ship across state lines because we've been doing it. But the good thing about hemp is that 0.3% is a lot. In terms of instrumentation, instruments can see so much lower than that. So you can make tons of standards at 0.3%. So there is something to that, to be able to do a round robin even at that level, to be able to show even on a difficult matrix that laboratories can see similar results or not. And then based off of that information, be able to know exactly where you need to target for improvement.
I think there's also a technical difficulty in doing a round robin. It's that basically having a single reference material that's fully homogenized and can actually be shipped across state lines, it's really hard. Like, how do I find a butane hash oil sample with exact same amount of butane, and how can I ship that to other labs to test among my results? So it's definitely going to be helpful, but it's definitely challenging right now.
I think the round robin testing, or ring tests, as I'm used to calling them, is definitely a really good idea. The thing that we're seeing, at least within certain states, like I know that recently there was a publication out in the journal Nature that took a look at all the Washington state labs and took a look at the potency results, and you saw quite a bit of variance between the labs, and you could always pinpoint, where a certain result came from. And I think that comes down to, well, how is the sample being prepped? How is the instrument being calibrated? What instrumentation are you using? And being able to see, the distribution of how people report their results is definitely very important. And I think that should be implemented. That was just a sample of Washington State cannabis testing laboratories, but I think something like that should be implemented across nationally to take a look at, how are states performing between each other and who's reporting the more accurate result.
As an internal auditor, I would say yes, do a round robin. The standard for ISO says for in-house developed methods, you either have to partake in proficiency testing or interlab, intralab proficiency testing, where appropriate. So going for accreditation, you really have to plan this out if you're doing round robins, especially with your accrediting officer, for them to understand that this industry does have a lack of resources like reference materials. We are strapped with our hands around our back when it comes to shipping samples, even within state. And so it has to be very robust, and for proper validation, do not skip out on proficiency testing no matter how you can do it.
We have 1 minute.
Okay, so my question is it's more of a suggestion, I would say.
You had talked about MIPS and how they come in all different forms, whether they be gummies, candies. I mean, basically now they're putting it into anything they can find. Why not test the starting product, whether it be the oil or what have you that goes into the MIP instead of the final product? So then you don't have to develop all these different methods for all these different matrices. If you were to test what's going into it at the start and then know if it's contaminated, it's free of pesticides or microbials. Just
That's one way we could do it, and then you would have to have a two-part testing set up, right? So you could test the oil for contaminants, but then in the final product, you still have to go back to testing for potency.
Yeah, and then you don't actually know if the other ingredients that they're putting in it is also safety tested. So either way, you'd probably have to test it again. I mean, there could be pesticides in the non-cannabis ingredients they use, which is something we've seen a lot in other products.
It's as somebody who works for a company with our vertically integrated labs, it's important to test from conception of a product all the way to the end result, especially when you're getting materials from vendors, questionable vendors, China and whatnot. It's important to test them in the beginning. You go through such a rigorous process of getting to your gummy bears or whatnot, which does require heat and require a lot of manipulation. It's advantageous to monitor your whole process through manufacturing and production using objective evidence by testing at every step to see if there's a part here you need to troubleshoot. But at the end of the step, especially depending upon which state regulations require you to also provide information on the final product. And so it's important to not only do beginning and end, but advantageous to do throughout the whole process.
Thank you very much.